Proteins are basically the hard-working employees of a cell. Each employee has an important role or function within the company, but to this day, we actually still don't know what many of these employees are doing. This is a problem because most of the drugs that we take rely on a solid foundation of knowledge about the proteins in our body. We do have some information, including ideas and suspicions about most proteins, but some are multifunctional, meaning they're involved in a lot of different cellular processes. So how can we study proteins and figure out what they're doing in our cells? Like any normal company, proteins work together as a team, forming networks and interactions with one another. So in a normal company, to glean information about an employee of interest, you can see which other employees they're working with. If you have some idea what the others are doing, you can make an educated guess that your employee is involved in that process as well. This field of research is known as proteomics, studying groups of protein and their function. And today I'm going to talk a bit about my PhD work. In the past, researchers were able to identify a handful of candidate proteins, represented here as black dots, that interact with a specific protein of interest, seen here as an orange dot. With this information, we could learn a limited amount about the protein of interest. Now, in the present, thanks to a huge 2012 advance in technology, researchers are able to identify all the proteins that come in close proximity to a specific protein of interest. This technique is called BioID. My PhD thesis relies on me using the BioID technique and its newer models to identify candidate proteins that interact with my multifunctional protein of interest. Using this technique, I was able to identify new networks, processes, and complexes that may involve my protein, vastly increasing our ability to study its essential role in the cell. The point that I really want you to walk away with from this is that thanks to this technique, we can now study protein network and function collectively. And because of this technique, we've changed the way proteomic studies are conducted, allowing us to see the bigger picture of what's happening inside our cells. Thank you.